My name is Sumon Pradhan. I am an R&D scientist at Thermo Fisher Scientific Cell Biology based in Carlsbad, California. Today I am going to talk about the molecular assays that we offer for a comprehensive characterization of stem cells, specifically pluripotency, differentiation potential, and genomic stability. At the end of this presentation, I hope you would appreciate that our assay methods significantly reduced the effort and challenges in this space. To begin with, I would like to give a brief introduction on stem cell workflow and the characterization requirements. Shown here is the schematic of stem cell workflow. iPSCs are generated from somatic cells via reprogramming. Cells are then expanded and either edited or differentiated into multiple cell types. With the advent of new methods and technologies in the past few years, we as scientific community have made significant progress in regenerative medicine, utilizing stem cells as a tool for the treatment of many complex diseases. As stem cells move from bench to bedside, there is an imperative need for extensive characterization methods. While characterizing stem cells and their derivatives, along with maintaining pluripotency and differentiation potential, it is also critical to confirm that the cell line has not acquired any genomic abnormalities following derivation, extended passaging, or genetic manipulation. In addition to that, it is important to uniquely ID every parental and subsequent derivative cell line, especially useful for core facilities, stem cell banks, or clinically relevant cell lines that require high level of traceability. This slide summarizes the traditional method of checking pluripotency and lineage-specific differentiation of stem cells using differential enzyme dye, surface markers, and intracellular marker staining using a host of antibody dye conjugates. For example, self-renewal marker expression is confirmed by alkaline phosphatase live stain or other markers like SSEF4 and TRA60 or OCT4 in undifferentiated cells. CD44 serves as a negative cell surface marker. On the other end, lineage-specific marker expression in differentiated cells, for example, TUS1 for ectoderm, smooth muscle actin for endoderm, and alpha-fetoprotein for endoderm by intracellular staining. Although the antibody-based staining method is easy to use, but these are a bit subjective, and one of the major drawbacks is low throughput. They are not scalable. Here comes the molecular methods. We have three molecular assays, one for confirming pluripotency, one for trilineage differentiation potential, and the third one for assessing genomic stability. First, an array-based global gene expression assay for confirming pluripotency. The assay is compatible with PluriTest, an established method with over 16,000 samples analyzed to date. It is cost-effective, the analysis is free and simple to use and cloud-based. Second, TACMAN PSC Scorecard panel. This is a quantitative assay confirming both pluripotency and trilineage differentiation potential. The results are compared with an extensive set of reference standards. The assay is easy to use, comes as a pre-plated assay and intuitive analysis software. The third one, KaryoStat assay, an array-based alternative to G-banding karyotyping. It is cost-effective and makes accurate detection of chromosomal abnormalities. Simple analysis with no cytogenetics expertise required, and turnaround time is around 3 to 4 days. First, I'll talk about PluriTest. PluriTest is a bioinformatic assay for pluripotency. This is the closest alternative to the teratoma assay based on global gene expression data. The assay compares the transcriptional profile of a sample to an extensive reference set with over 450 cells or tissue types, including 223 human ESCs and 41 PSC lines, and it provides instant pluripotency analysis results. Pluripotency confirmation utilizes two separate scores, pluripotency score and novelty score. A pluripotent cell is often characterized by a high pluripotent score and a low novelty score. One of the major benefits of this array is multiple independent measurements per transcript. For example, we have 11 probes per set for well-annotated and 9 probes per set for the not so well-annotated genes. As of now, the array offers the highest transcript coverage in the industry with most recent annotation. The array has about 50,000 probe sets, and multiple controls have been integrated into the workflow that allow easier quality control checks. This slide summarizes reagents, instrumentation, and analysis portal to carry out this assay. There are two array formats available: individual array or a plate containing 16 arrays, a freely downloadable software transcript analysis console for QC of the scanned files, and an online cloud-based portal, PluriTest.org, for the analysis. Bottom right panel is the instrumentation required: a fluidic station for wash and stain the chips, and a scanner. GCS 3000 for prime view array, and Gene Titan for 16 array plates. This is the assay workflow. Poly controls are added in the beginning that helps tracking the entire workflow. The assay starts with the first and second strand cDNA synthesis. Next step is the in vitro transcription coupled biotin labeling. Follow it purification, fragmentation, and hybridization to the array. After washing off non-specific and excess probes, chips are then scanned. The assay utilizes an online cloud-based analysis software to get the results. Now a bit details on the analysis process. Up the left is the actual web page for PluriTest analysis. Analysis requires uploading cell files, and it outputs the results in a format as shown on the right. Top panel states whether the sample is a pass or a fail. Bottom panel is a 2D plot, pluripotency on the y-axis and novelty on the x-axis. The two parameters based on a pass or a fail is determined. Here are the PluriTest assay results from a number of different cell types and cell states. For example, on the left panel, we have a bunch of undifferentiated ESCs and PSCs that are clustered at the very top. Pluripotency score decreases with a simultaneous increase of novelty score as stem cells undergo differentiation. Compare mid-differentiation dopamine neurons and transplant stage dopamine neurons with ESCs and PSCs. Similar trend follows as depicted in the panel to the right. Day 7, 14, and 21 embryonic bodies have low pluripotency score and high novelty score as compared to iPSCs. HeLa and fibroblast serve as completely differentiated cells. Now I'll talk about the second assay, TaqMan PSC Scorecard. The assay contains 93 genes sensitive to early germ layer specification, including self-renewal, ectoderm, mesoderm, and endoderm, including controls. Together, the assay assess trilineage differentiation potential of new or established pluripotent stem cell lines. Key benefits include a very simplified workflow, a standardized assay tested across different labs, culture conditions, and genetic backgrounds. And the assay is well accepted in the scientific community. What you see on the right is a box plot representation of an undifferentiated ESC clone which displays only positive score for self-renewal and negative score for 3 germ layers. This slide summarizes the assay workflow and instrumentation. The assay consists of 4 major steps: RNA isolation, cDNA preparation, qPCR step in a preplated Taqman assay, and finally the data analysis that utilizes a free online cloud-based software. Preplated assays are available in 96-well as well as 384-well format and are compatible with a wide range of qPCR instruments. Here is an example showing how scorecard analysis can be used to assess the different cell states as stem cells undergo differentiation. Embryonic bodies were generated and cultured in suspension culture over the course of 14 days prior to scorecard analysis. The summary shows that the ESCs, which was only self-renewal positive in the beginning, can generate all 3 germ layer specific cells. For example, EB formation at day 2 is positive for ectoderm but negative for mesoderm and endoderm. As EB formation progresses from day 4 to day 14, the scores for all 3 germ layers continue to increase with a concomitant decrease of self-renewal genes. The table below with the scores corroborates the same pattern. Red denotes upregulation and blue denotes downregulations. Here is another example of characterization of an undifferentiated iPSC clone with day 7, day 14, and day 21 EBs. Bottom panel shows the differential plot for day 0 iPSC clone versus day 7, day 14, and day 21 EBs. As evident, the clone continued to express more of ectoderm, mesoderm, and endoderm specific genes with the simultaneous decrease of self-renewal genes as they undergo differentiation. Now, the third assay, KaryoStat and KaryoStat HD, that assess the genomic stability of iPSC clones. The assay provides an alternative to G-banding karyotyping and also offers genotyping with whole genome coverage for accurate detection of chromosomal abnormalities with results in just 3 to 4 days. It is cost effective. The analysis is simple with no cytogenetics expertise required. Here is the most conventional method of cytogenetic analysis, which is G-banding. The assay requires growing cells, arresting them in metaphase, fixing and spreading them on a slide, and staining with Giemsa. G-banding detects gross chromosomal structural changes, deletions or duplication at the resolution of 5 to 10 megabase. The assay takes at least 2 weeks to get the result. Small deletions or duplications cannot be detected because of its low resolution power. Most importantly, data analysis and interpretation requires experienced or trained professional. Here is what we offer and the side-by-side comparison of KaryoStat and KaryoStat HD with other methods used to assess genomic integrity. Currently, we offer this assay in 2 formats depending on the resolution. Low resolution format, KaryoStat, is designed for basic research, whereas high resolution format, KaryoStat HD, is geared more for translational research. In addition to features that G-banding offers, our assays offer absence of heterozygosity and allelic information based on SNP probes as well. Now a bit more on the array. The array utilizes a strong reference and probe sets. The reference has been built based on over 300 normal individuals and the cell lines. We have about 315,000 features for KaryoStat and 2.6 million for KaryoStat HD. A significant of these features are actually SNP probes. One of the biggest advantages of SNP probes is that it allows detection of allele frequencies that in turn enables a more sensitive detection of mosaicism, independent confirmation of copy number state, and better breakpoint determination. As evident, the array includes genes recommended by international standards for cytogenomic arrays, Mendelian inheritance, red-seq genes, and the intergenic backbone. This slide summarizes the kits and the instrumentation required to carry out this assay. The assay requires exact same instrumentation as PluriTest, a fluidic station for washing and staining, and a scanner to scan the chips, GCS 3000. Analysis software CHAS, which is freely downloadable and open to all. Here is the assay workflow. The assay needs about 250 nanograms of genomic DNA. It starts with digestion with a restriction enzyme, followed by an adapter ligation for the subsequent PCR step. PCR product is purified. The next step is the fragmentation, which is the most critical step in this assay. Therefore, a mandatory QC gel is necessary, and ideal fragment size should be between 25 and 125 base pair. The fragmented DNA is labeled with biotin and next hybridized to the probes on the array. Non-specific DNA is washed off and then stained before scanning the array. Shown here is the karyo view of a good ESC clone with no chromosomal abnormalities detected. Karyo view is the digital view of individual chromosome 1 through 22, X and Y. There are two vertical lines that parallel to each chromosome. One line displays the copy number gain or loss. The other line is for mosaic copy number state. Any chromosomal loss or gain will be reflected on those vertical lines as red or blue triangles depending on the chromosomal loss or gain respectively. This slide shows that KaryoStat assay results are consistent with G-banding results. We derived two iPSC clones from a parental fibroblast. G-banding confirms that both the parental line and the derived iPSC lines are normal and female cells. So does the KaryoStat assay, as there is no chromosomal abnormalities detected. Cyan lines represent the parental line, orange and green lines represent iPSC clone 1 and 2 respectively. And this is how a karyotypically abnormal clone, which is trisomy for chromosome 12, 17 and X, would look like in our assay. KaryoStat assay detects two things: trisomy for chromosome 12, 17 and X as depicted by solid deep blue bars, and mosaic copy number gain for the entire chromosome 12 and 17 as detected by light blue bars. Both panels shows the detailed view of chromosome 17. As I briefly mentioned, we also offer high-resolution version of this assay, KaryoStat HD. Panel on the left is a karyo view of a good iPSC clone with no chromosomal abnormalities detected, whereas KaryoStat HD detects a chromosomal loss on chromosome 7. Bottom panel shows the description of the chromosomal abnormalities. It is located on chromosome 7 The size of the loss is 132 kilobase, and this region does not contain any genes. In cases some genes are associated with the region, the software tells the number and the name of the genes. To summarize how the newly launched molecular methods PluriTest and KaryoStat align with our long-term goal of providing a more comprehensive characterization in combination with the existing products such as Scorecard. One complements the other. PluriTest confirms pluripotency by an exhaustive analysis of global gene expression profiling, whereas Scorecard utilizes a focused gene panel, tells us about the lineage-specific differentiation potential and pluripotency. And finally, KaryoStat performs karyotyping and genotyping in one single experiment. With that, I end my presentation. Thank you.